you ever wondered, what are the most important papers in the field of evolutionary biology? What are the studies that were so influential that they fundamentally changed the way we view the science of evolution? Today, we're going to do a top 10 countdown of the most important papers that every evolution enthusiast should have on their reading list. Hello everyone and welcome back to the channel. If you're new here, I'm Zach Hancock, an evolutionary biologist who specializes in population genetics. Regular viewers of this channel will know that I'm super into the history of evolutionary biology, and like all sciences, the field has been shaped by a series of highly influential studies that today form the foundation of evolutionary theory. Many viewers have emailed and commented asking questions like, if I want to know more about evolution, what are some important books or papers that I should read? And so it seemed to me like there may be an audience for a video of what I consider the most fundamental papers of the field. Now, I should caveat here that these are what I consider the most important. A different evolutionary biologist might have their own top 10 list, but for each I'll explain exactly why I picked them. I'll also add that it was very difficult to narrow it down to only 10, so I'm going to include just a couple of honorable mentions in the description, though there are probably a ton more that are equally as important that didn't make the list. I did, however, try to come up with a semi-objective way to form this list, and what I decided upon was citation number. For all the papers on this list, they must have been cited at least 1,000 times. This means that some really important modern papers didn't quite make the list because they fell below this threshold, often just due to their recency. So, most of the papers are older, which also means that they tend to be dominated by white men, since white men have unfortunately held a disproportionate position in science for most of its existence. I'm going to do these in order from 10 down to 1, with number 1 being what I think is the most important paper in evolutionary biology. This too was very difficult, and I wouldn't put too much weight on the ordering itself. Okay, without further ado, let's get this top 10 countdown of the most influential papers in evolutionary biology started. Coming in at number 10 is a paper published in 1999 by Alan Force, Mike Lynch, and friends in the journal Genetics, titled Preservation of Duplicate Genes by Complementary Degenerative Mutations. This paper has been cited over 4,000 times, despite being the youngest paper on this list, and for good reason. This paper introduced the concept of subfunctionalization, an extremely important idea in the evolution of new genes. Historically, we thought that new genes emerged first by duplication. A single gene becomes two initially identical, and then the daughter copy, over time, mutates and takes on a new function. This was known as neo-functionalization. However, this process is relatively rare, and instead most duplicate genes degrade and become pseudogenes. This led researchers to believe that duplicate genes shouldn't last very long. On evolutionary timescales, they should be purged from the genome. But that's not what they found. Duplicates persisted for a long time, even when mutations had clearly disrupted their original function. Why? What Alan Force and colleagues found is that when duplicate genes exist, both the duplicate and the original can accumulate mutations. This is because the duplicate can compensate for mutations in the original and vice versa, freeing both of them to mutate. When this occurs, the parent and the daughter copy become reliant upon one another. This fixes them both in place. Now a function that once only needed a single gene to be performed needs two. This also generates the beginnings of a gene interaction network between them. Subsequent duplicates can elaborate this network even further, and indeed can provide the basis for increasing molecular complexity. Since Force et al., we've learned that most complex molecular machines like ribosomes and spliceosomes evolved in this manner, by initial duplication by mutations that cement the duplicates in place. They called this process subfunctionalization to distinguish it from neofunctionalization, and dozens of studies since have shown that their mechanism is far more common in nature than neofunctionalization. Okay, moving on to number 9, we have Jerry Coyne and H. Alan Orr's highly influential paper titled Patterns of Speciation in Drosophila, published in Evolution in 1989. For many, this paper serves as the founding of the modern field of speciation genetics, that is, studying how new species form by looking at their genes to identify signatures of reproductive isolation and divergence. What Coyne and Orr did was sequence genes from many species of a genus of fruit flies. Some species are what are called sister species, meaning that they are one another's closest relatives, while others are more distantly related. What they asked was a very basic question. Are closely related species more likely to hybridize than those more distantly related? They were interested in the ability to hybridize because a key component of the biological species concept is that good species should fail to reproduce or produce viable offspring in nature. But since speciation is a continuous process, not something that happens instantly, then they reasoned that reproductive barriers were likely to be incomplete the more closely related you were. While this makes intuitive sense today, Coyne and Orr were the first to show it empirically in fruit flies. As predicted, closely related species were more likely to have weaker reproductive barriers than those that were more distantly related. But they went further. A major question in speciation research is how quickly these barriers emerge. A prediction is that species in allopatry, that is, those separated by a geographic barrier, form reproductive barriers more slowly because there is no selection against hybrids, since they are never in contact with one another and so can't form hybrids. Alternatively, if two species are in sympatry, they occur in the same area and might occasionally hybridize, selection should be stronger against hybrids. This idea was first proposed by Alfred Russel Wallace and is known as reinforcement. And indeed, Coyne and Orr found exactly that. In allopatric taxa, reproductive isolation was much weaker than in sympatric taxa, regardless of how distantly related they were. Many studies have been performed since, mostly clarifying and supporting these original conclusions, but the paper was the first to test these long-standing theoretical ideas with an empirical system, and thus deserves a place on this list as one of the most influential papers in evolutionary biology. Coming in at number 8 is Russell Landy and Steve Arnold's groundbreaking paper from 1983 titled The Measurement of Selection on Correlated Characters, published in Evolution. With nearly 6,000 citations, this paper is truly a giant in the field of quantitative genetics. For most of the history of genetics, mathematical models have been constructed assuming that traits were independent of each other, and that natural selection could act on each of them in an independent fashion. We've always known that this wasn't true, but no one sat down and worked out the math and actually derived what would happen if natural selection was acting on traits that were actually correlated with each other. Landy and Arnold did just that. They derived a simple mathematical model composed of the product of two matrices. The first, called the G matrix, is a matrix of genetic variance on the off-diagonal and genetic covariance on the diagonal. This covariance captures how two traits can be correlated with each other. The matrix is then multiplied by a vector of selection coefficients acting on each of these traits. What's useful about this approach is that selection can be trying to optimize traits in different directions, and depending on how much covariance they share, the underlying genetics of the traits can constrain how selection is expected to behave. For example, when there's no genetic covariance between traits, selection moves a trait through a fitness landscape in the most straightforward way, quickly reaching an adaptive peak. However, when traits are correlated, selection does not take the most straightforward path. It follows a curved trajectory through the adaptive landscape. Their model has been expanded to include not only genetic covariance, but also phenotypic covariance, which has allowed paleontologists to reconstruct a G-matrix even for extinct taxa. In fact, when there exists a time series of fossils from a given population, using Landy and Arnold's model, paleontologists can recreate ancient adaptive landscapes and follow how those populations tracked the changing landscape through time. Their model also has important applications in agriculture and livestock breeding, as it helps to clarify how farmers should select certain traits depending on how they're correlated with each other. For all the theoretical and empirical advances the Landy and Arnold model has provided, as well as real-world applications, it deserves a special place on this list. At number 7, we have Josh and Esther Lederberg's 1952 study titled Replica Plating: An Indirect Selection of Bacterial Mutations, published in the Journal of Bacteriology. While the title doesn't sound all that flashy, the study is immensely important in establishing the role of mutation in evolution. I want to start with a bit of a biographic note. Josh Lederberg would go on to win a Nobel Prize for the discovery of bacterial conjugation and generally gain widespread acclaim, while his wife, Esther, would not. Despite the fact that she discovered the first lambda-phage virus in bacteria, devised the entire technique of replica plating, and even worked on bacterial transduction, she would not even be given a tenured faculty position in her lifetime. At least according to me, she's the co-author of one of the most important papers in evolutionary biology, and yet during her lifetime was barely treated as a respectable scientist because of her gender. Okay, now on to the paper itself. For much of the history of evolutionary biology, there have been lots of debate about the role of mutation and the environment in shaping variation. Darwin believed that the environment induced beneficial mutations whenever the species needed it. Others, like August Weismann, disagreed, believing that there was a barrier between the germline and the somatic cells that prevented direct environment intervention and adaptation. There was no theoretical answer to this question. It had to be resolved in a lab. So the Lederbergs took up the task and devised a brilliant little experiment to test the idea. They started off with an agar plate where they let bacterial colonies grow. Next, they used Esther's method of replica plating. They stamped the colonies from the original plate onto a replica plate that was then treated with penicillin. While most of the colonies on the replica plate died, some of them were resistant to the antibiotic and survived. The question is, did these colonies evolve resistance in the presence of the penicillin, or was their resistance just a part of the standing genetic variation they possessed before being exposed to penicillin? That is, did the environment induce an adaptive mutation, or did the mutation come about randomly without respect to the original need? To find out, they treated the original plate with penicillin. It turned out the exact same colonies that were resistant to the antibiotic in the replica plate were resistant in the original colony. This meant that that resistance had emerged randomly prior to the exposure, not because of the exposure. Otherwise, you would not expect the exact same colonies to be resistant in the original plate. This study was one of a few that established the evolutionary principle that mutations are random with respect to fitness. You don't get a beneficial mutation just because you need it. This is a textbook idea at this point, but the Lederbergs were one of the first to demonstrate it. Moving on to number 6, we have our first paper that has been cited over 10,000 times. This paper was penned by paleontologist Stephen J. Gould and population geneticist Richard Lewontin, titled The Spandrels of San Marco and the Panglossian Paradigm, a critique of the adaptationist program, published in 1979. Unlike the previous papers on this list, this one is mostly a thought piece. It's an artistic but biting critique of the way evolutionary biology was being performed at the time. This past summer, I had the pleasure of visiting the Cathedral of San Marco in Venice, and given the immense importance of this paper, had my own kind of religious experience standing beneath the vaulted ceiling and the golden spandrels. This paper is ultimately a critique of what is called adaptationism, the idea that every trait that exists is there because it is adaptive to the organism. They invoke Pangloss from Voltaire's Candide, who famously says that our nose is so designed so that spectacles can sit on it. Voltaire was critiquing Leibniz's idea that we live in the best of all possible worlds. For Gould and Lewontin, the critique in the study of evolution has produced the best of all possible phenotypes. They use the spandrel as an example. If you want to build a curved ceiling, you must include arches. Because arches are a structural necessity, there will exist the parts connecting the arches called spandrels. Often, architects will decorate these spandrels with beautiful murals and sculptures, which might lead some to conclude that the spandrels exist so that there can be beautiful paintings on them, when, in fact, they are purely a structural necessity of an unrelated part. A great deal of the paper attacks what they call just-so stories. These are adaptive stories that we tell that whenever we want to make sense of a trait without having any evidence to actually support that story. For example, what if I asked you, what is the adaptive purpose of men growing beards? You might say that, well, men fight a lot, so men that grow thick beards might be more protected against being punched. So, natural selection favors men with thick beards. I use this example because, amazingly, a study tried to argue exactly this. An alternative, of course, is that men grow beards because they have more testosterone for completely unrelated reasons to fighting, and beards are just a side effect of producing that hormone. They are a spandrel. Regardless of your feelings about Gould and Lewontin's critique of adaptationism, and there are many who quite disliked the paper, you can't deny how incredibly influential it has become, to the point that spandrel, an obscure architectural term, has now permanently entered the biologist's lexicon. Okay, now, halfway through our list, coming in at number 5 is John Maynard Smith's highly influential paper, The Theory of Games and the Evolution of Animal Conflicts, published in the Journal of Theoretical Biology in 1974. This paper has been cited over 3,000 times and is in many ways a follow-up paper to the one that Maynard Smith and George Price of the Price equation fame wrote on the mathematics of animal conflict. While not technically the first paper to use game theory in evolution, it was the one that sort of formalized its use. Game theory comes from economics and is concerned with strategic interactions, conceptualizing the interactions between two things, whether supplier and consumer or predator and prey. Maynard Smith imagined trying to generate a mathematical prediction of why certain behaviors might persist in a population. For example, in many social organisms, there is a sharing of resources. There are also, however, cheaters who refuse to share. If natural selection is favoring individuals, then you might expect that all cheaters will do better than sharing their resources because the cheaters always get all the resources for themselves. However, using game theory, Smith showed that this is not always the case. Depending on how that behavior is coded, there should actually be a stable balance between how many cheaters exist and how many sharers there are. This is because cheaters don't contribute to each other, and so as their frequency starts to rise, they start to do worse as a strategy. Cheating, very often, is only good when there are more sharers than there are cheaters. Whenever an equilibrium can be reached between two behavioral strategies, Maynard Smith called this an evolutionarily stable strategy. He found that not all behaviors were this way, and so some behaviors must be evolutionarily transient. One of the most famous games he developed is called the hawk-dove game, which investigates the evolution of animal conflict. He showed that under certain circumstances, fleeing from a fight and losing a resource is actually better than fighting and keeping some component of that resource if, in the long run, you incur more damage than benefit. This paper and Maynard Smith's follow-up book on the topic is seen as having birthed the field of behavioral ecology, bringing mathematical modeling to the evolution of animal behaviors. It's rare to say that a single paper represents the birth of an entire field of science, but this next paper, coming in at number 4, is exactly that. published in 1918 by R.A. Fisher. The paper, titled "The Correlation Between Relatives on the Supposition of Mendelian Inheritance, is Responsible for the Entire Field of Quantitative Genetics." Not only that, but it demonstrated mathematically that continuous traits could be completely explained by discrete Mendelian genes, something that was still hotly contested in the early 1900s. This paper is one of those that is extremely dense, and rarely anyone actually reads it, but it's full of so many important insights that it simply had to make the list. Apart from unifying continuous traits with Mendelian genetics, it also introduced the concept of heritability, which Fisher measured as the correlation between parents and offspring in trait value. You can think about it like this. For a trait that is entirely determined by genetics, children are expected to have the exact same trait as their parents. They are perfectly correlated, and hence have a heritability of 1. Whenever the environment is also involved, then there is noise introduced that causes children to deviate from their parents, making the correlation less than 1. For a trait with a correlation of 0.7, for example, means that 30% of the variance is caused by the environment and 70% is caused by genetics. Fisher also shows that heritability has an important impact on the strength of selection. He demonstrates that natural selection is constrained to shift the mean trait value of a population by how much traits are correlated between parents and offspring. That is, natural selection needs genetic variation to function, not just environmental variation. Further, he showed that natural selection, when it's acting on the mean of a population, doesn't care about dominance or epistatic effects of genes. It only cares about the additive genetic variance. This is the variance that is captured by a linear regression, as would be performed when searching for the correlation between parents and offspring. Hence, all that other stuff, genetic and non-genetic, only acts to slow selection. This insight is profoundly important and serves as the basis of the breeder's equation, which has been used in livestock and agriculture for 100 years at this point. Indeed, one of Fisher's main interests was in how we can use the principles of natural selection and genetics to improve crop yields. While it may be one of the hardest papers on this list to actually read, it has been cited over 6,000 times, and its influence on the field of evolutionary biology is undeniable. Alright, coming in at number 3 is the most cited paper on this list at 22,000 citations. It's W.D. Hamilton's 1964 paper titled "The Genetical Evolution of Social Behavior, Part 2, published in the Journal of Theoretical Biology. As stated, this is the second part of a two-part paper in which Hamilton introduces the concept of kin selection. While the first part is highly mathematical and rarely cited, the second is more conceptual and easy for biologists to digest. In fact, splitting this paper into two parts was a suggestion that Maynard Smith, who served as a reviewer on this paper, made to Hamilton. Hamilton was seeking to answer one of the biggest mysteries in evolutionary biology, the evolution of altruism. The concept deeply troubled Darwin, who wondered how natural selection would ever favor a system in which some individuals did not breed. He tried to argue that the answer was in group selection, a form of hierarchical selection that favored the entire colony as a reproductive unit. But many population geneticists showed that group selection was highly unlikely. Instead, Hamilton started from the idea that if there was a genetic component to altruism, and that altruistic acts somehow could favor the proliferation of individuals that shared your genes, then natural selection could in fact favor it. Since the probability of sharing genes decreases the more unrelated you are, altruism was most likely to evolve among close relatives. He called this kin selection. The simple expression is RB greater than C, where R is the coefficient of relatedness, B is the benefit to the recipient, and C is the cost to the altruist. For the trait to evolve, the inequality must be satisfied. Notice that even when the benefit is large, if you are virtually unrelated, then it's still extremely unlikely that for altruism to to evolve. Kin selection has wide support in natural systems. Most organisms that demonstrate altruistic behavior do so among close relatives, and when altruistic behavior is absolute, as in the case of self-sacrifice, it is almost exclusively done among clones. For example, the slime mold is a congregation of many individuals that form a stalk, in which only those at the top survive and reproduce, while all the individuals along the stalk die. Why would they ever choose to play this role? Turns out slime molds are clones, so the genes of individuals in the stalk still get passed on so long as those at the tip survive. Without a doubt, Hamilton's kin selection theory is one of the most iconic and recognizable of evolutionary theories for a popular science audience, and I'm sure most of you will have heard of it. Hamilton made many more incredibly important contributions, including resolving the evolution of spite, the opposite of altruism, but kin selection theory is definitely the most influential. Alright, here we are folks, nearing the end of our list, and this one was very difficult to decide, but I think this placement is correct. For number 2, I'm choosing Moto Kimura's 1968 paper, Evolutionary Rate at the Molecular Level, published in the Journal of Nature. This paper is unique in that if you were to read it devoid of historical context, you might not think too much of it. But in 1968, this paper caused a firestorm and set off what would become known as the naturalist selectionist debates that to some extent continue to this day. In the early 1960s, we started to get the first ever measurements of how much genetic variation there exists in nature. Most biologists at the time believed that natural selection either worked to remove harmful variation or to quickly fix adaptive variation. This meant that there shouldn't be a ton of variation between individuals. But what these early studies found was that there was a ton of genetic variation. Some people tried to argue that this variation was either on its way to fixation or that it was under balancing selection, such that the heterozygotes were more fit and so selection was maintaining all that variation. Kimura, relying on an argument first made by J.B.S. Haldane in 1956, showed mathematically that this could not be the case. If selection were favoring that many variants, it would create such a substitutional burden on the population as to drive it to extinction. Instead, Kimura argued that most of that variation was selectively neutral. It had no effect on the fitness of the organism. Kimura immediately recognized how important this insight was. First, he noted, if this is the case, then genetic drift, not natural selection, may be the most important evolutionary force acting at the molecular level. Next, if this was true, then the molecular divergence between two organisms is independent of their population size, and thus can tell us how long ago they split. This concept became known as the molecular clock, which had been pointed out previously, but lacked a theoretical mechanism for its existence. Lastly, Kimura showed that standing levels of variation, if shaped by drift instead of selection, could serve as a proxy for the size of the population. Because of this, the field of conservation genetics was born. To this day, we use how much genetic variation a population has to calculate how big that population is. Small populations have less genetic variation than larger ones, an insight that only makes sense under Kimura's view of evolution. While the term neutral theory of molecular evolution wasn't used in the paper, this is indeed the paper that jumped off neutral theory. From neutral theory, a ton of subfields were born, including molecular phylogenetics, conservation genetics, molecular ecology, and many others. It became possible to test for selection at the molecular level because now we had an expectation of what regions should look like if there was no selection at all. In effect, Kimura provided us a kind of null model of evolution. So how neutral is evolution? Is genetic drift really the dominant force over natural selection? The debate rages to this day, and the question likely has no simple answer. Instead, some organisms, like large-bodied vertebrates with historically small populations, like ourselves, are probably more influenced by drift, whereas others, like bacteria, protists, some insects, etc., are probably shaped more by natural selection. Kimura would spend the rest of his career writing about and defending his neutral theory, and it remains to this day the only real contender to dethrone Darwinian evolution, that is, evolution by natural selection. Because of this, Kimura joins the pantheon of great evolutionary biologists, and this paper is what catapulted him to this position. Alright folks, it all comes down to this. My pick for the #1 most influential paper in all of evolutionary biology. My guess, before revealing it, is that this pick might come as a surprise because I suspect the vast majority of you have never even heard of it. But in the case of this paper, I actually think that's a strength because the concepts of this paper are so ingrained in evolutionary biology that people rarely even cite the paper anymore. Each of the concepts of this paper have become literally textbook knowledge that we have forgotten where it originates from. So my #1 pick for the most influential paper in evolutionary biology is Sewell Wright's 1931 paper titled Evolution in Mendelian Populations, published in the Journal of Genetics. This paper has been cited over 12,000 times, is 63 pages long, and basically serves as the reference for all topics in classical population genetics. In it, Wright reviews what we today call Hardy-Weinberg equilibrium, followed by detailed derivations of each of the forces of evolution: selection, drift, mutation, and gene flow, and how they emerge naturally from violations of Hardy-Weinberg. He introduces the concepts of equilibrium, both between selection and mutation, migration and mutation, etc., which foreshadows work by Haldane on the genetic load and Kimura on mutation drift equilibrium. He introduces textbook concepts for the first time, like effective population size, how gene frequencies are expected to fluctuate under drift alone, the inbreeding coefficient, and even, for the first time, provides a general formula that captures all the forces of evolution in a single expression. It is also in this paper that he introduces one of the most important metaphors in all of evolution, the fitness landscape. Wright envisions this landscape as a three-dimensional topographic map with peaks and valleys of fitness, where each position in the map represents certain combinations of genes. Using his general formula of evolution, he tries to predict what sorts of populations would move readily through this landscape. While he doesn't develop it fully in this paper, he does speculate on the fact that subdivided populations in which drift is partitioned between subpopulations might cross valleys more readily to find new peaks than in populations that are really, really large and randomly mating. This would become known as the shifting balance theory, and it's one of Wright's biggest theoretical contributions to evolutionary theory. In many ways, Wright's 1931 paper represents the culmination of the preceding decade's work in theoretical population genetics. It represented a massive shift from organismal and comparative biology to a sort of mathematized biology, one based on laws, theorems, and statistics. This shift was made possible thanks to the rediscovery and acceptance of Mendelian genetics, leading Wright to conclude, quote, "Any theory of evolution must be based on the properties of Mendelian factors, and beyond this, must be concerned largely with the statistical situation in the species." Wright, in this 1931 paper, saw himself as providing a unified theory of evolution based on the principles of Mendelian genetics and the mathematical situation of the species. So there you have it, my top 10 picks of the most influential papers in evolutionary biology. As a reminder, all of these were cited over 1,000 times, and some were cited over 10,000 times, but are otherwise my own personal preferences. You will likely have noticed my bias towards papers in mathematical evolutionary genetics, but hey, that's the part of evolutionary theory I engage with the most. As a reminder, I've also included a few honorable mentions in the description box. So what do you think? Did I miss a paper that you think should have been included? What would be your top 10? Let me know in the comments below. Thanks so much for being here. If you enjoyed this video, let me know, and maybe I'll do a top 10 books on evolution next. Hit like and subscribe, and anyway, thanks so much for being here, and I'll catch you all next time.